Thank you very much. It's really a pleasure to be able to be here. Can you tell me that you can hear me?
Yeah.
Can you give me a thumbs up? Great. I'm really very deeply honored to be able to be here to talk to all these young Ukrainian scientists. When I was first asked to present this lecture, my first reaction was, but you Ukrainians need so much more than a lecture from me. I thought of all you've endured and how my words are so small in comparison. And yet here you are, and you're looking to the future, and there's so much to be hopeful for. I'm happy to be here to tell you about the fun of curiosity-driven science. I want to celebrate both your curiosity and your desire to make a difference. It's very fortunate that with science, you don't have to choose between your own interests and helping others. You can do both. You can pursue your own curiosity and at the same time, make a difference in the world. I'm going to tell you a story about a discovery that I made by following my curiosity and the unexpected impact that this discovery had on medicine. I also want to highlight that scientific advances don't come from just one person, one idea, but rather many people collaborating allows full understanding to unfold. So I will tell you the story of discovery and highlight some of the people who made this possible. Is this slideshow working?
Yeah, everything.
You can just nod. Great. So what I'm going to tell you about are the ends of chromosomes. Chromosomes are made of DNA. So within all of your cells, you have the cell nucleus. And in this nucleus, there are many chromosomes. And each one of those chromosomes, as shown here, is made up of DNA. The DNA consists of specific base pairs. So we call this A for short, and T, these base pair with each other, and C and G. And the order of these base pairs encode for everything that the cell has to do. And so this is a blueprint for what the cell carries out. In addition to all of the DNA that is coding along this chromosome, there are special kinds of DNA at the very end of the chromosome that we call telomeres. So if you look here within the cell nucleus, each one of these chromosomes, all of the encoding information is along the length of the chromosome, and the telomere plays this very special role. The telomere has to protect the chromosome end, and it also has to maintain the length of the chromosome. And I'll be telling you what I mean by that as we go through this talk. Telomere was first defined by Barbara McClintock in 1943. She was interested in what allows chromosomes to function, and she defined this protective function for the ends of the chromosome. It wasn't until 1978 that we understood what the DNA sequence was at the end of this chromosome. And we now know that the telomere sequence was identified as a very simple tandem repeat DNA sequence. So shown here, TT, GGGG, TT, GGGG, TT, GGGG. And this was identified in this single cell pond animal shown here called Tetrahymena thermophila. And the reason that Joe Gall and Liz Blackburn were able to identify this tandemly repeated sequence in this organism is that this single-celled pond animal has 40,000 chromosomes. So if you're interested in some particular aspect of the cell, you go to the cell that has the most of that to try and be able to study it. And so this is why this single cell tetrahymena was used to try and understand the DNA sequences at the ends of chromosomes. Now, this DNA sequence doesn't code for anything like the rest of the genes along the length of the chromosome, but rather it provides a protective buffer zone at the end of the chromosome. So this is Joe Gall and Liz Blackburn when they first identified the sequence of the telomeres as these tandemly repeated sequences. So we know that the telomere is this repeated DNA unit. Here is my cartoon of the chromosome, and the telomere are these little blue ovals here, which are these repeats on the ends of the chromosome. And the way that a chromosome is copied every time a cell divides, it turns out that the mechanics of how that copying occurs doesn't copy to the very end of the chromosome. So every time you make a copy of a chromosome, there's a little bit of telomere repeats that are lost from the ends of the chromosome. And so we've known about this problem replicating the end of the chromosome for many years. And the question is then, how could it be that we could all be here with our chromosomes intact if we're actually losing sequences from the end of the chromosome every time cells divide? And it turns out that there is this very clever machine in the cell, an enzyme called telomerase, which helps fix this problem of the telomeres shortening every time the cells divide. So the telomeres shorten during cell division, so you have fewer of these repeat units. But this enzyme that we call telomerase adds telomere repeat units back onto the chromosomes. So this is my little cartoon. We'll see a little bit more about telomerase in a few minutes. This is the cartoon of the telomerase working very hard to put repeats back onto the chromosome ends. So thus the chromosome is a buffer at the end of the chromosome, the telomere. There's some shortening during cell division and then some lengthening due to telomerase. So there's constant shortening and lengthening of the chromosome ends. And this was the discovery that I made when I was working with Liz Blackburn, because we were curious. We were just curious, how is it that the ends of the chromosomes are maintained? So this was me back in 1983 on the hunt for telomerase, trying to understand how it is that chromosome ends are maintained. And the way that we did this was to utilize, again, this single-celled pond organism, Tetrahymena. We took many of these cells and we made a little cocktail, a little biochemical cocktail out of these cells. And we put this into a tube that had a little bit of a telomeric repeat. You can see this is 3 repeats. And it had a building block of the TTP and the GTP that have to be added to make the telomere repeats. And this GTP was labeled with radioactivity so that we could follow this in the experiment. So we have this single-stranded telomeric oligonucleotide, which is indicated just in this arrow here. And then we have the building blocks, the G and the T. And what we found is that this cocktail of this Tetrahymena cells, there was this enzyme, this machine in the cell that could take these building blocks and add onto the end of this oligonucleotide the GGGTT, GGGTT. And you'll notice the beautiful colors of this, this telomere repeat here. And the way that this looks, when you look at this in the cell, is this repeating 6-base ladder on a sequencing gel. And so when we first saw this repeated ladder on the sequencing gel, that was a real aha moment. Wow, we had found something completely new that people hadn't understood before. And this is one of the exciting things that one can do in science, is just to find something new. We now know that the telomerase enzyme is a very complex and interesting enzyme. I won't have time to talk to you about all of the biochemical experiments that went into understanding telomerase, but we know that it has an essential RNA component that we call here telomerase RNA. And you can see this dark gray here represents the structure of that telomerase RNA. And within the RNA, there's a region that contains a template. You can see that template region right here. The template region is also surrounded by the catalytic protein component of telomerase that we call telomerase reverse transcriptase, or TERT for short. And so together, the components of this enzyme are able to add repeats onto the ends of chromosomes. This is Julian Chen, who was responsible for understanding the structure of that telomerase RNA that I showed you when he was working with me in the lab. And what we understand now is here is this telomerase machine here, and here is the end of the chromosome. And the template region of the telomerase RNA will allow sequences to be added onto the chromosome end, and then the enzyme can move and add some more sequences, and then move and add some more sequences. So it functions by copying an internal template. Again, this was very exciting to understand how this enzyme actually carries out its function. So I mentioned that with every cell division, the telomeres shorten a little bit, and then telomerase, as we just saw, can add sequences back on. And so if you look at all of the chromosomes in the cell, and that's what's shown here, here are the telomeric repeats. Each one of the chromosome ends in the cell has a slightly different number of those telomeric repeats. So if you have a way to cut all of those chromosome ends, and then look at the length of each one of these telomere repeats, what you find is that there's an equilibrium length distribution. And what I mean by that is that there are a few telomeres that are very short, a lot of telomeres that are medium length, and a few telomeres that are very long. So the telomerase enzyme will maintain this equilibrium length by, when telomeres get shorter, adding repeats back on. And the big question that we are trying to understand right now is the regulation of telomerase and how is this length maintenance occurring. Carla Connolly was a scientist who worked with me for over 20 years trying to understand this length maintenance of telomeres and by regulating telomerase. So why does telomerase matter? It's interesting and it's fun, but why am I here talking to you about telomerase and what it has to do with disease? It turns out that by continuing to follow our curiosity and understand telomerase in cells, that we found that telomerase is required for all cells that divide many times. So one example is during normal tissue renewal. You have cells in your body that are called stem cells that are necessary for the renewal of tissues. And these cells can make a copy of themselves, but they also can give rise to specific tissue cells that carry out specific functions in your body. And in order for the telomeres to be maintained during these many rounds of cell division, telomerase is absolutely essential. The other area where telomerase plays a role is in cancer. You have these stem cells that are making copies of themselves, but they also give rise to these specific differentiated cells. And if there's a change that happens in one of those cells that allows that cell to divide more times, it can go on and form a tumor by dividing many, many times. And again, telomerase is essential to maintain the telomeres for these tumor cells to be able to divide many times. So I'll tell you a couple of stories about both the normal tissue renewal as well as cancer. And again, this started with a curiosity-driven question. We wanted to ask, what happens to cells if they don't have telomerase? So this was when I first started my lab and was asking these questions. By becoming an independent researcher, it really allowed me to follow my curiosity to find out what is the next thing that we really want to understand. And again, science is a team effort. This was our group trying to understand why telomerase matters. So in order to answer this question, we used mice, because mice can help us understand the telomere function. The question we wanted to ask was, what happens if telomeres can't be elongated? So we tested a mouse that lacks the telomerase enzyme. So we used specific technology. A number of years ago, it was different, but there's a way that you can take a mouse and be able to knock out a specific gene. So we knocked out the gene that encodes the telomerase RNA component, and then tried to understand what happened to those mice. And when we generated a mouse, if you focus here, this first generation mouse we call G1. This is the mouse where we first knocked out the telomerase component. And that mouse was perfectly fine. There wasn't anything wrong in that generation. But when we took this G1 mouse and crossed it to another G1 mouse, we then would get the second generation mouse, or G2. And we saw that the telomere repeats in this G2 mice were slightly shorter than they had been in the G1 mice. So then we took these G2 mice and crossed them to each other to generate the third generation telomerase knockout mouse. And again, in this generation, we found that the telomeres were slightly shorter. We took those third generation mice, crossed them to each other to generate the G4 mouse, and found that the telomeres were shorter. And we were able to do this breeding up until just 6 generations, and then we couldn't breed any further due to the complications due to short telomeres. And I'll be telling you about that in just a minute. What we found is that the short telomeres cause a loss of tissue renewal capacity. So in the blood, we found that there was bone marrow failure. In the intestine, there was a loss of the cell lining. In the skin, there was decreased wound healing. In the hair, there was premature graying. And in the testes, there was cell death and infertility. And after 6 generations, we couldn't breed these mice any longer. And this is Margaret. All of the studies that we did on telomerase knockout mice would not have been possible without all of her expert mouse genetic skills. What we found is that the telomere length equilibrium has a threshold. So I mentioned that normally telomeres are regulated around this equilibrium. So if this is all of the telomeres in the cell in the telomerase knockout first generation telomerase null mice, that equilibrium shifts to shorter telomeres in the late generation telomerase null mice. And we discovered that there's a threshold, and that when telomeres get to be below this threshold, then the cells stop dividing. So they either undergo programmed cell death, or they simply stop dividing and senesce. And so the short telomeres cause the loss of the tissue renewal capacity. So telomere shortening leads to cell death or cell senescence. So here we have these telomere repeats on the ends of the chromosome, and with each round of DNA replication, the telomere repeats shorten from the end of the chromosome. And when they get to be critically short, then they cause cell death. So the telomeres are a buffer zone. They play this protective role at the end of the chromosome, but it's only when they get to be very short that there's a problem. And that's why our first generation, second generation, and third generation telomerase knockout mice didn't have any problems. It wasn't until the telomeres got to be short in the 4th and 5th generations that there was a loss of tissue renewal capacity. So these short telomeres cause either cell death or cellular senescence that leads to the tissue degenerative effects. And this is Mike Heeman, who was a graduate student in the lab, he's now a professor at MIT, and he really was instrumental in us understanding that it's the shortest telomere that causes the effects in the telomerase null mice. So I've told you that telomerase is required for cells that divide many times. And we were still curious about how telomerase plays this role in normal tissue renewal and moving from mice into human cells. We and others showed a number of years ago that telomere shortening occurs in normal human cells. And there's a wide distribution of normal telomere length in the population. So what's plotted here on the y-axis is the telomere length. And this is the age of individual people. And this is looking at white blood cells. And so you can see that there is a wide range of telomere length in the normal human population, and that the telomeres shorten progressively with age. So we were very interested and curious to know what role this telomere shortening plays in human biology. We found that inherited mutations in telomerase cause a disease termed telomere syndromes that mimic age-related disease. So this is a family which my colleague Mary Armanios studied a number of years ago that came into the clinic at Johns Hopkins with this individual here having a bone marrow failure syndrome. And you can see from this family, what we see here is the grandmother and grandfather, and then the son, another son, another son, and a daughter. And those individuals that are shown here in black had mutations in telomerase so that they didn't have enough telomerase enzyme activity. And you can see that the later generations, this individual here had bone marrow failure and was the first one that we studied in this family. And by collecting many such families, we now can understand what happens in humans when they have insufficient telomerase activity and telomere shortening. And what Mary and others were able to find is that these individuals have bone marrow failure, pulmonary fibrosis, a lethal lung disease, immunosenescence, emphysema, chemotherapy intolerance, liver cirrhosis, and gastrointestinal disease. Many of these effects are very similar to what we had seen in our telomerase knockout mouse. So this is a case where by following our curiosity, we had first studied the mouse, and then when the human disease became apparent, it was very clear that the telomere shortening with the increasing generations is what's giving rise to this, because the knockout mouse had already been studied. Typically, one would do this in the opposite direction. That is, one would have an inherited human disease, and to be able to understand that, one would make a mouse to mimic the human disease. In this case, we had made the mouse 10 years before and had already studied it. So we were a little bit ahead of the game in understanding this disease. One of the things that occurs in these families is that with each generation that the mutation is present in these families, that there's a progressive severity of disease with each generation. And again, that's precisely what we saw in the telomerase knockout mouse. So we could understand this progressive severity of disease by the work that we had done previously in the mouse. And this is Mary, who now is continuing to work on these inherited diseases at Johns Hopkins, has a registry with over 100 families where we can really understand the biology, the human biology of the disease. So in addition to the normal tissue renewal, telomerase is essential for cells that have to divide many times. And we were also curious about the role of telomerase in cancer. We know that telomerase is activated in many cancer cells. Telomerase is not active in most human cells, although it is active in some stem cells to allow for this tissue renewal that we've been discussing. 90% of human cancers have activated telomerase. The other cancers use an alternative method to elongate telomeres. I won't be able to go into that now, but all tumors have to solve the problem of maintaining their telomeres in order to continue to grow. So maintaining telomeres is essential for cancer growth. David Feldzer was a PhD student in my lab who really focused on understanding the role of telomerase in cancer, and he's now a professor at University of Pennsylvania. So we understand that there are consequences for telomeres that are both too long and too short. So we have this telomere length distribution. Telomerase, shown here in green, can elongate the telomeres, and then DNA replication shortens it. And the importance is to maintain this length distribution. There's a short telomere threshold, and if the distribution falls below this line, then we have stem cell failure and a loss of tissue renewal capacity and degenerative disease. There's also a long telomere threshold, and if telomeres are continuously maintained, there is a predisposition to cancer. So it's really critically important to understand this distribution of telomere lengths. How is telomere length equilibrium established? What is the molecular mechanism? This is the question that is really driving my curiosity right now. We know that we have telomeric DNA, and that telomeric DNA has to interact with telomerase. You can shorten the telomere with replication, and telomerase will elongate it. But how is it that this maintains the equilibrium? How is this process regulated? And this is the question that we are continuing to study right now. So I'll just summarize what I've told you. I've told you that telomerase is essential for telomere maintenance. Telomere shortening leads to cell death after many cell divisions. Short telomeres limit the growth of cancer cells. Short telomeres limit tissue renewal and contribute to age-related degenerative disease. So my take-home message is that curiosity-driven research generates unexpected discoveries that give insight into human disease. When we set out to understand the telomerase in the single-cell pond animal Tetrahymena, we didn't know that there was a connection to pulmonary fibrosis and cancer. And it was only by continuing to follow our curiosity to understand how this works that we were able to uncover these really important links to human disease. So before I go on and give credit to the people in the lab that have done this, I wanted to take a minute to talk about my colleagues here at University of California, Santa Cruz. And the Genomics Institute here would like to be very supportive of Ukrainian scientists. My colleague, Olena Vaska, has put together some assistance if displaced scientists would like to have some help from the UC Santa Cruz Genomic Institutes. There will be free temporary storage of genomic data if you need that, temporary computational resources, a free bioinformatics immersion program, 1 to 2 day hands-on bioinformatic training. And if you want to express interest in any of these opportunities, you can email this email address here, displacedscientistgroup@ucsc.edu. There's a lot of people that would be very happy to provide any help that we can have. So just giving credit to my lab, this is when we dressed up as chromosomes. You can see the chromosomes here. And following our curiosity, we just have fun doing science. And then I would just like to tell you about, this is an example of curiosity-driven science and its impact on medicine. And there's really a revolution going on in science right now that's spurred by new technologies. There are all kinds of opportunities opening up for young scientists. The revolution in my field of biology is really astounding. The rate at which we can generate new data is very hard to comprehend. The new tools in genomics and microscopy have led the way. There's also a revolution in structural biology with computational approaches to understand protein structure that was unheard of even 6 months ago. But how do we make sense of all this data? We need your curious minds. Using science, your interest and talents can make a change. There's so much to discover. Being trained as critical thinkers, we scientists can look behind the main story and see the truth. And I'm happy to have you on that path with us. So thank you, and I'm happy to answer any questions if there are any.
Thank you very much. It's for a great lecture. It was a great pleasure to hear you today, to be with you today. Yeah, we have a little questions. Does one of the reasons of a biological death is the inability of telomerases to fix the telomerase shortage?
So, telomerase plays a role in age-related degenerative disease. It's not the only thing that determines the maximum lifespan. So we know, for instance, that mice that have long telomeres or mice that have short telomeres, they still have the same maximum lifespan. So there are many different components that contribute to maximum lifespan, but the telomerase does play a major role in age-related degenerative disease, the immunosenescence and the pulmonary fibrosis that I talked about. And of course, age-related degenerative disease will shorten lifespan of individuals. So there is a component to that.
All right, the next one, does the telomere shortage weaken the immunological response of the body?
Yes, one of the effects that one sees in individuals that have not enough telomerase is an immunocompromised state. So the immune system requires the telomerase to maintain telomeres to be able to function efficiently.
Great, great. And the last one question, like, tell us a little bit about your experience at the Nobel Prize ceremony, how it was, what did you feel like that?
Yes, it was quite an honor to get the phone call that I had won the Nobel Prize, and that set off a whirlwind of activities over a number of months, and the way that the Swedish academies and the Swedish people treated those of us that were there for the Nobel ceremonies was really quite remarkable. There was a whole week of activities, meeting young scientists, giving talks, hearing from my colleagues. It was really the honor of a lifetime to be able to participate in that with my colleagues.
Thank you very much for a great lecture. I hope that very soon we will meet in Kiev, and maybe you will make in-person lecture in some of our big universities.
I would be honored to be able to visit Kiev in the future.
Thank you very much, and have a good day.
Thank you.